Hi everyone, my name is Savariar. I am a Pharmacogenomics instructor at Manchester University College of Pharmacy in Fort Wayne, Indiana. I received my Bachelor of Science degree in chemistry from Huntington University, and I received my Master of Science degree in Pharmacogenomics at Manchester University College of Pharmacy. My job consists of teaching and research. So for teaching, I teach lecture courses, which are also called didactic courses, and I teach Introduction to Pharmacogenomics, which gives you, like it says, an introduction to what pharmacogenomics is. And just in short, pharmacogenomics is a mix of looking at a person's medications and their genes to find out what medications work the best for them. This is really a cutting edge field of science because I'm sure you guys can think of someone that you know where they took a medication and you've heard them say, hey, this medication didn't work for me or it made me sick. And that's exactly what pharmacogenomics is looking into, is looking at each person as an individual and not just as a mass, and looking at how different they are and seeing that different medications work for them. So we're actually able to isolate a person's DNA and look at their DNA and see the variations that run, and then eventually end up looking at genotyping reports, which I'll show you later on. So you can do everything from getting a swab sample to running the DNA sample through some tests, and then getting an end product, which is like a genotyping report. So you can look and see what medications that you are okay to take, that maybe you should have caution with, and medications that you shouldn't take. So this is really useful because it helps people feel better. Anytime that you feel sick, you want to know that you're taking the best thing for you. So instead of taking a medication that could actually make you feel more sick or not help you at all. And that's exactly what pharmacogenomics is. For the skills necessary, you do need to have a little bit of research experience or laboratory skill experience. So if you are interested in going in a science field, you will be getting that science laboratory experience in your classes. So whether it be a chemistry class or a biology class, all of those things will help you for a pharmacogenomics lab. But when you get to this program, we teach you everything from picking up a pipette to actually generating a genotyping report. So at the end of the pharmacogenomics program, you actually feel like you are enabled and able to do all of these things. In a typical day, I usually do research and teaching. So for teaching, I teach the didactic, which is also called a lecture course, and I also teach laboratory courses. So for both of those, it's a little bit different. For lecture courses, I usually wear a business casual outfit, and I usually either present to my class or we have discussions, and I lead the group and make sure that my students are understanding what they need to know. For laboratory courses, it is my job to make sure that people understand the hands-on techniques, because if they want to work in a lab for like pharmacogenomics research, or they want to work for a pharmaceutical company, they need to know certain techniques. And we teach them everything from isolating DNA, so taking a cheek swab, and you can isolate DNA, you can isolate RNA. We show them how to analyze on gel electrophoresis or Western blots. We do a little bit of cell culture. We can actually take your DNA and then basically isolate it with a really cool machine called a Kingfisher, and it uses magnetic binding beads to basically take up the DNA, and then it washes them, and you get pure DNA at the end of it. You can get your concentration of DNA, but you can also do a lot more quantitative things with your DNA, like find out what variability that you have. Right here, this is actually one of our techniques that we show our students how to do. So this is called a genotyping chip. So basically, these are consisting, so each block or each square right here has 64 holes, And what that does is once you isolate your DNA, we use a robot to fill this machine with DNA sample, and each little hole right here has an assay or a gene variation stuck to it, so you can identify what type of variation that you have. And once we run this, we use a PCR technique, so it's called polymerase chain reaction, where you can amplify your DNA. You can actually identify what variability that you have in your genes. And what that looks like is it actually ends up looking like a giant stack of papers it's called a genotyping report. So basically, so I have a sample report here to show you. So what this looks like is it shows you, for example, like if this person is more likely to be depressed, if they have high cholesterol, it shows you what medications that they can take and what medications that they shouldn't be taking. So for example, this person here, these are all the medications for antidepressants that they are okay to take. This shows you use with caution, and this shows you medications that they should not be taking. So this is in conjunction in working with a physician and a pharmacist. So a lot of our students are in our Master's in Pharmacogenomics program, so they're actually isolating the DNA samples and running these genotyping reports. They can also do COVID testing, for example. They can do all sorts of things with the techniques. So we learn PCR, Western blotting, but it's really useful for when you get into a lab because you have hands-on experience. And for pharmacists, this is really helpful because pharmacogenomics is really closely intertwined with what they do every day, because they have to deal with prescribing people medications and working with the physicians in order to do so. So looking at pharmacogenomics helps pharmacists better able to prescribe the right medication. And it helps people from having adverse side effects, which means they're not going to feel well, or worse, that they actually end up in the hospital because they have a medication that made them ill. So some of the other skills that I find really valuable in my career is being very meticulous, and I always have to keep up to date with material and learning, especially because pharmacogenomics is such a new and growing field. So I have to always keep up to date and make sure that I have the newest knowledge in order to teach my students the information that they need to know to actually go out and make a difference. But also in the laboratory with new techniques, or just practicing in order to show better technique to my students, and just making sure that I'm learning so I can teach them something new. But also being meticulous in the way that I keep my my lab. So when I run experiments, just making sure that everything is properly labeled. I'm wearing the correct like protective gear, so lab coat, gloves, eyeglasses, mask, and making sure that I'm not contaminating everything, or just making sure that everything is right where it's supposed to be, so I can get the best results at the end of the day. So for my career in pharmacogenomics, so if you guys are interested in learning more about pharmacogenomics, or you think that you would be interested in it, if you're the type of person that always likes to learn and wants to help work in healthcare, or like help better people's lives, then and be on the cutting edge of science, then this is definitely for you. So I would really consider looking into what pharmacogenomics is. Thank you.